If we're seeing this, it's already too late. This is the naked capsid of the hepatitis B virus, moments after it has infiltrated a normal liver cell by hacking a run-of-the-mill gate on its surface. For hundreds of millions of people, this represents the point of no return. Hep B's capsid is now being rapidly transported to the nucleus of this cell, where it will break apart and unleash one of the most intense takeover programs in nature. In a world where most viruses try to live fast and transform an entire host cell into a rapidly dying capsid bomb, hepatitis B opts to take a slower, clumsier approach, stumbling its way to gradually converting this liver cell into a viral factory, co-opting cellular export machinery to infect other liver cells, and to deploy chaff that confounds the immune system. Hep B is a barely optimized infectious agent, relying almost entirely on this liver cell's metabolism to assemble new viral particles. And yet somehow, this sloppy strategy has resulted in hepatitis B being one of the deadliest infections of all time, touching the lives of nearly 2 billion of the people on Earth today, with up to 4% of the entire human population saddled with a chronic, near-incurable hepatitis B infestation. Today, let's explore the life cycle of one of our deadliest opponents and see how the stealth approach shatters our defenses. Let's get into it. And hey, welcome back to Clockwork, a show about the molecular foundations of life. Subscribe so you don't miss new episodes as we explore the base mechanics that root you to the world. And make sure you check us out over at patreon.com/clockworkshow to see our perks like monthly sticker packs and our great molecular papercraft series. But we've got a lot to cover here, so let's not waste any time. In this video, we're going to loosely follow the life cycle of the hepatitis B virus, from entry to Hep B's incorporation into the nucleus to the wild and complex assembly and export process for new virus particles. Doing this is going to show us the strange, stumbling path Hep B has taken to world domination. But let's cover the ultra basics. Hepatitis B is a virus, a kind of genetic aberration that exists to repurpose your cellular machinery to build more copies of itself. Hep B's quote-unquote goal is to smuggle its own very weird DNA code into cells in your liver. Despite the fact that hepatitis B only spreads through direct contact with bodily fluids, it is remarkably resilient and infectious. And here's the headline. According to the World Health Organization, there are over 200 million folks suffering from some form of chronic hepatitis B infection, with estimates that 2 billion people, over a full quarter of the world's population, have been exposed to the virus. And those same WHO estimates pin over 1 million deaths per year on hepatitis B. But the numbers are fuzzy because those deaths come from liver failure and the staggering amount of liver cancer that hepatitis B can cause. Only our old nemesis tuberculosis can really edge out those numbers, and just barely at that. The molecular biology we're about to explore will help explain why the burden from Hep B is so high. So let's just jump right in. If you're going to actually wrap your head around how hepatitis B works, you first need a quick primer on its point of entry, the hepatocyte, or liver cell. Hep B enters via the bloodstream and is perfectly structured for infecting your liver cells specifically. Since all roads lead to the liver eventually in our circulatory system, it's a great vector for infection. More importantly, these surface hepatocytes are just primed to be the perfect partner for hepatitis. Your liver cells are very active, constantly pulling metabolites or harmful chemicals out of your bloodstream and then processing those chemicals for whatever their next step is. Your liver is a dizzying factory floor and water treatment facility all bundled into one organ. Hepatitis B is going to exploit a lot of these processing pipelines to quietly turn this hepatocyte into a long-term virus factory. And here's how it starts. This is the full hepatitis B virion. You're looking at the outer membrane of the virus. This outer membrane protects a little protein shell, more on that later, but there are also these little protein nubs embedded in the membrane, the hepatitis B surface antigens. Unlike a lot of other viruses, only a very small part of the surface protein actually pokes out of the membrane. This little nub is like a lockpick, and it fits perfectly into receptors on the surface of our hepatocyte here. These receptors are meant for pulling bile salts out of your bloodstream, but HBV can activate and hitch a ride on this exact conveyor belt. A bunch of proteins work together to bring the hepatitis B virion into this liver cell once this receptor gets activated. And I really need to emphasize how this liver cell is going to automatically do most of the work here in terms of establishing and maintaining a long-term hepatitis B infection. For real, hepatitis B is one of the most stop-hitting-yourself infections I've ever studied. Once inside, hepatitis B will lose its outer membrane, exposing the core protein capsid of the virus. The Hep B capsid is just along for the ride at this stage. This is just around 150 to 200 copies of a single protein assembled into this spiky sphere. The capsid protein is just the right shape needed to establish this infection. Each copy of the protein has a little tag on the end of it, a small string of protein called a nuclear localization signal. Again, essentially, hepatitis B doesn't need to do anything once it gets inside. The moment it enters, it will bump into and get picked up by a group of proteins called importins, which bind to that nuclear localization signal and then automatically pull hepatitis B's capsid down into the nucleus. These importins also create a shield that allows this whole little complex to be pulled across the nuclear pore, one of the most well-defended gates in any one of your cells. Hep B is going to get an all-access pass here essentially for free, which means now hepatitis B can get down to business. Once inside the nucleus, the Hep B capsid will break apart, but the exact details of how that goes down are poorly understood. Regardless, breaking down the capsid reveals the actually infectious core of hepatitis B, an incredibly weird and broken circular packet of viral double-stranded DNA. I cannot emphasize enough how bizarre hepatitis B's genome is. This is a roughly scale-accurate simulation of hepatitis B's DNA packet. Hepatitis B is only packing 3.2 kilobase pairs of DNA here, 3,200 letters of DNA, which is basically nothing. That much DNA information is only enough to encode like 2 copies of your hemoglobin protein, and the average intron of non-coding DNA in your genes is, no joke, double the length of the entire hepatitis B genome. But quick fun fact, this incredibly small DNA packet only makes hepatitis B the second smallest viral genome that infects humans. What's the number 1 smallest virus, you ask? Hang tight, we'll actually get to that by the end of this video. So, Hep B's genome is super small, but look carefully. Turns out, Hep B's DNA also arrives broken and incomplete. You've got this intact double-stranded section here attached to a long stretch of single-stranded DNA that's still attached to the viral polymerase protein that built this busted structure in the first place. And by this part of the infection cycle, my jaw is already on the floor. So far, hepatitis B has effectively done 0 actual work, prepared a vanishingly small DNA payload, and arrived with a half-busted genome in order to take on and defeat one of the most robust and multi-layered immune systems known to the natural world. The difference in scale here is astounding to me. Hep B is basically packing a spork to take on and defeat an entire aircraft carrier. But keep watching, we'll see why the system is secretly brilliant. Because your cells simply cannot tolerate the presence of broken DNA of any kind. Instead of purging this dumb gambit from hepatitis B, your own DNA repair mechanisms are going to take over here and quote-unquote fix this broken double DNA strand, completing Hep B's genome for it. And I mean, incredibly, these repair mechanisms will even go so far as to package and process the hepatitis B genome using your own histones, turning an incomplete viral genome into a mini chromosome. This covalently closed circular DNA packet is now ready to just pretend it's part of your genome and get to work quietly building virus parts. So many other viruses come in guns blazing, completely hijacking cellular metabolism and blowing up their victims to spread as many copies of themselves as possible, as quickly as possible. Hepatitis B is here to take things slow, to slip in through the cracks and pull off a gradual takeover, which is going to cause so many downstream problems. And those problems, according to this recent paper, are kicked off the moment this mini chromosome forms, because in this section of the mini chromosome right here, the liver cell's own machinery will predictably add histones in this exact same spot, which will guarantee that DNA transcription proteins will attach and start assembling viral RNA from Hep B's smallest protein coding section. This is critical because this tiny 465-letter segment of DNA codes for hepatitis B's ace in the hole, the appropriately named X protein. This is effectively the only tool in hepatitis B's offensive arsenal. What you're looking at here is a simulation of the X protein, as the shape of this thing is honestly very loose and hard to pin down. The X protein is here, in short, to cause pandemonium. Most of this little glob is inherently disordered, but it can cohere into a few different shapes based on the other proteins it is close to. This allows X to shapeshift its way into disrupting nearly 200 known proteins and pathways in human cells. One weapon, hundreds of targets. You could almost get away with describing X as a molecular cluster bomb. Protein X can have far-reaching effects like this because it's more of an adapter protein. It lightly redirects cellular metabolism by bringing different complexes together. And let me illustrate that with one of X's most important jobs, which needs to happen right now if Hep B has any hope of gaining a real foothold in this hepatocyte. X needs to be the first hepatitis protein on the scene because the whole smuggling viral DNA into a human nucleus thing really shouldn't work out, like not even a little. Because you've got a whole army of proteins patrolling the nucleus to make sure that no foreign DNA has infiltrated this inner sanctum. We've evolved tons of nucleus-based countermeasures to ensure our cells are only doing what they're supposed to do. Managing your genome is one of your most important jobs, and if you fail at that job, you either get sick or get cancer, so it's a huge deal. And Protein X is going to immediately start blowing that up by first binding to this DNA repair protein called DDB1. Proteins like DDB1 help with DNA repair, but it also helps detect faulty proteins inside the nucleus, working with cellular garbage disposal systems like this E3 ubiquitin ligase. Anyway, one side of X will bind to DDB1, and then the other side will attach to a central component in this beefy DNA maintenance complex called SMC56. And now that these two systems are in contact, the ubiquitin complex will just automatically tag SMC56 for destruction, despite the fact there's nothing wrong with this complex at all. So, as more and more X protein makes its way into the nucleus, more and more SMC56 will get destroyed. But okay, why is that bad? What does this SMC John do that's so important? SMC56 is a chromatin maintenance system, one of many that helps clean up and protect your DNA. SMC56's specific job is to recognize and bind to any suspicious or viral DNA it finds, blocking it from getting transcribed. So, sure enough, blowing up this antiviral defense protein is going to open the floodgates and allow hepatitis B's mini chromosome to start producing viral proteins unchecked. This hepatitis X protein messes with DNA maintenance in so many more ways, but the upshot is that X interference will disable viral countermeasures while also increasing the number of transcription factors that are recruited to the hepatitis B mini chromosome, which ensures that this liver cell is constantly producing more hepatitis B parts no matter what. But I guess that begs the question, what exactly can hepatitis build with such a limited genome anyway? And you know what, I've gone too far with the limited language here. It's better to think of hepatitis B as one of the most efficient DNA systems that can infect a person. Most of hepatitis B's genes actually overlap, so the virus is getting a ton of mileage out of a compact and easy to propagate DNA sequence. In total, there are 4 places where DNA transcription machinery can bind to the Hep B mini chromosome. We've already seen what the shortest one does with the X protein. In total, these 4 binding sites can build 5 unique mRNA strands, which can, through the magic of molecular biology, yield 7 different proteins. This is the full hepatitis B toolkit right here. And let's break down everyone's roles real quick and see how this all plays out. Hepatitis B's biggest need is a viral polymerase protein. This will get packaged inside the virus and help complete the assembly process by transcribing a long RNA back into hepatitis B DNA. Then we have the capsid protein, which will help form the main structure of the final virion. On top of that, there's these surface antigen proteins, which can come in 3 distinct flavors. And now the longest mRNA transcript is technically just here to act as a template to rebuild hepatitis B's DNA sequence in new virions. But if there's a small mistake and the transcription machinery binds around 30 letters upstream, then that long mRNA will eventually get assembled into this goofy variant of the capsid protein, the Hep B E antigen that comes with a little bonus tail. So all in all, we've got a copy machine, 2 structural scaffolds with some variations, 1 tiny agent in a pure biochemical chaos, and 1 mistake. That's all you need if you want to impact the lives of over 2 billion people on Earth today. So hepatitis B wins when these structural proteins self-assemble around a full copy of that pre-genomic mRNA attached to the viral polymerase. But hepatitis B doesn't encode for anything that helps push liver cell metabolism towards assembling Hep B virions. Right now, at the start of things, Protein X is just blowing up enough DNA maintenance systems to guarantee that the hepatitis B mini chromosome will pump out as many copies of these structural proteins as possible. Beyond that, hepatitis B just goes with the flow, hoping that enough capsid proteins will bump into each other to allow the virion to self-assemble. And I've said it before, but this borderline Zen playbook is going to have incredible downstream consequences. But right now, let's just assemble 1 virion and see how this is supposed to play out. Remember, this long pre-genomic mRNA strand is all we need to make a new copy of the hepatitis B genome. This mRNA will meet up with the viral polymerase, and capsid proteins will gradually self-assemble around this genetic payload. After the capsid is complete, the polymerase will activate and use that pre-genomic mRNA to build the half-complete hepatitis B DNA strand that's going to infect the next cell down the line. Meanwhile, these surface antigens really hate being out in the open cytoplasm and will either clump together or embed themselves in whatever membrane they can find. Most likely, they'll end up concentrating in this liver cell's endoplasmic reticulum. Now, all we gotta do is put these 2 main components together. Ideally, this freshly complete viral capsid will bump into a section of endoplasmic reticulum that's chock full of HBV surface proteins, which will just surround that capsid and butt off the membrane, completing our freshly new hepatitis B viral particle, which is called a Dane particle, by the way. I never mentioned that, but just want to have that out there. But this is rarely how things work out at first. I can't emphasize this enough. Hep B relies almost completely on normal liver business to assemble itself. The main thing this capsid is built to do is infiltrating the nucleus. Remember at the beginning when I showed you that the capsid proteins have a nuclear localization signal baked into their structure? That hasn't changed, and it can't change. So right here, when the capsid is fully assembled and getting ready to envelop itself, this moment where hepatitis B can claim a quick victory and get out scot-free, nuclear importance will probably just pick it back up and pull it back into the nucleus. For me, this is the crux of the video. Hepatitis B doesn't really have a plan beyond getting into the nucleus, so a lot of the initial virions that get assembled are just going to end up right back at the starting point. When folks talk about viral infections, you hear a lot about plans and intention, and about hijacking cellular machinery to build new viruses. Hepatitis B is a great window into how haphazard all this is. Hepatitis B has bet everything on an efficient genome, and that sometimes makes the virus appear downright klutzy, even though this little fella has stumbled its way to touching the lives of a full quarter of the world's population. But back to this infection cycle, hepatitis B can still turn this quote-unquote clumsiness into a huge advantage, because, look, once this additional capsid makes it back into the nucleus, it will also break apart, allowing this copy of the hepatitis B genome to get repaired and turned into an additional mini chromosome. So, just in case the initial infecting virion can't synthesize enough X protein in time to completely shut down the DNA defense squad, there's now double the number of mini chromosomes in this nucleus, which will produce double the amount of hepatitis mRNA and proteins. And this process is just going to keep repeating over and over until a kind of equilibrium is reached. Eventually, there are enough copies of the hepatitis B genome stuffed into the cell's nucleus, and enough capsids are being produced that the nuclear import system gets completely overwhelmed. On average, there's usually about 50 copies of the mini chromosome in an infected nucleus, but sometimes that number can go up to 100 to 150. So hepatitis B has multiple backup copies of its genome in case one or more get damaged, and more hepatitis B DNA in this nucleus will increase viral mRNA and protein production. This is going to become the central pillars of hepatitis B's success. It doesn't matter that this infection cycle is inefficient at assembling and releasing new virions. If we flood this liver cell with more and more hepatitis proteins, we'll have a better chance of enough of them randomly bumping into each other to assemble complete and infection-ready virions. But of course, this comes with a lot of complications. First of all, there's going to be downstream effects that work in hepatitis B's favor. As these hepatitis B proteins flood into the cytoplasm, they're going to start messing with some cellular mechanics. Notably, a lot of these hepatitis proteins are going to automatically limit the frontline immune response that could blow up this burgeoning viral factory before things get out of hand. First up, the X protein subterfuge is going to extend into the cytoplasm, obviously, hampering all sorts of internal immune pathways. Notably, X disrupts the mitochondrial antiviral signaling system, or MAVS, which recognizes viral infections and activates the production of type 1 interferons. Interferons are one of our best frontline defenses against viruses. At a high level, these proteins activate RNA-destroying machines that completely shut down all protein production in an infected cell temporarily. They also signal to neighboring cells that there's an infection happening, which will help call in immune helpers like these natural killer cells, which will activate the self-destruct sequence in the infected hepatocyte. While there's lots of ways this cell can activate type 1 interferon production, disrupting MAVS is going to turn off a critical early warning circuit for this cell. Meanwhile, it's not just protein X doing all the damage out here. Some of hepatitis B's structural proteins also just happen to inhibit internal immune responses as well. And while there's dozens of examples, I don't want us to be here all day, so I'm just going to show you the one that blew my mind the most, because this is about the wildest thing I've seen in any infection cycle. Hepatitis B's surface antigens mess with the major vault protein. If you've been plugged into the life sciences space at all, you've heard about these massive, mysterious vault complex present in all of our cells. We aren't exactly sure what these things do, but there are lots of solid theories, and we're even using them to help study internal cellular mechanics. Based on studies focused on hepatitis B, we know that cells produce more of these vaults when a viral infection kicks off. And we also know that the major vault protein, or MVP, interacts with another immune signaling system, MYD88. But somehow, hepatitis B surface antigen proteins crowd around these vaults and prevent MYD88 from joining the party. Okay, cool, but who cares? What does the interaction between MVP and MYD88 actually do? And, it helps kick off type 1 interferon production. Nice. So, in short, hepatitis B's flood the zone strategy actually works out really well as it kneecaps the frontline immune response. However, this chaos does kind of cut both ways, and things don't work out perfectly for hepatitis B here, because the assembly process for these hepatitis B virions is actually incredibly inefficient. Sure, this cell is gradually pumping out complete infectious hepatitis B virus particles, but because there is zero control of this process, the cell is actually pumping out a lot more useless stuff. First up, the endoplasmic reticulum of infected cells is going to get so completely stuffed with hepatitis B surface antigens that empty little viral bubbles are going to spontaneously bud off and leave without any payload. The same thing happens with these capsid proteins. They'll self-assemble even without that pregenomic mRNA and polymerase inside them. and those empty capsids and empty surface shells will still get exported out of this liver cell. And the same actually goes for this misprint of the core protein, the hepatitis B E antigen. So while this hepatocyte is building and releasing infectious Hep B virions ready to infiltrate nearby cells, it's also spewing a bunch of non-functional Hep B detritus that seemingly does nothing. But of course, that can't be the end. How do you think hepatitis B is going to use this mess in its favor? Hilariously, all these quote-unquote wasted particles are going to act like a kind of chaff that overwhelms and almost literally exhausts the host immune system. Immune coordinators like T cells will get so overburdened by viral stimulus that they'll just stop responding to the presence of hepatitis B proteins, which can slow the immune response long enough to make a hepatitis B infection chronic. And I could really go on all day here, but these basic details illustrate the foundations of what makes hepatitis B so infectious and so pervasive. Sure, for most infections, hepatitis B will eventually get eradicated once the immune system gets its act together and mounts a coordinated enough response to overcome all the viral chaff and interferon silencing. But depending on the age a person gets infected, hepatitis B can infiltrate enough liver cells and exhaust the immune system enough that the infection becomes chronic and borderline incurable. Without a strong immune response, a huge percentage of the 250 million people affected by chronic hepatitis B don't have very noticeable symptoms until things start going completely off the rails. For one thing, the X protein's reign of terror will gradually lead to more and more genomic instability in these liver cells, leading to the development of severe liver cancers. Even without the cancer, a hepatitis B infection lives at the DNA level, so the immune system only has scorched earth options like apoptosis and severe inflammation to try and eradicate a Hep B infestation. After decades of chronic infection, this can cause profound liver damage that leads to liver failure. So for a lot of folks in the end stages of a chronic hepatitis B infection, a liver transplant is their only option. To me, hepatitis B is just a deeply humbling disease to learn about. Last year I took y'all on a deep dive into tuberculosis and how that bacteria had developed so many incredible, seemingly specific hacks that hamper the immune system enough for it to establish an infection. Hepatitis B is almost the exact opposite kind of problem, where our enemies can sloppily stumble through the cracks of our metabolism and gradually build up enough of a defensive front that we irrevocably become hepatitis B viral factories. Hep B is a critical reminder that biology is just the whims of chemistry and physics at the end of the day, and sometimes all you need is a few interlinked feedback loops to become incredibly dominant. But this haphazard and explosively inefficient infection cycle has one more astounding complication, the most mind-blowing one of all. Hepatitis B is so uncontrolled and so messy that this infection cycle actually leaves the virus open to other viral parasites, or satellite viruses that can only exist in the presence of a hepatitis B infection. In which, I mean, I cannot believe that something like that exists. Remember like a million years ago when I mentioned that hepatitis B has only the second shortest genetic code of any human-infecting virus? Well, hepatitis B directly enables the number one player, hepatitis D. This unbelievably short bundle of viral RNA and scaffold protein can only survive if it can envelop itself in hepatitis B's surface antigens. It doesn't code for them. This RNA just codes for its scaffold protein. And the way that this satellite virus works is just astounding. But here's the thing. Satellite viruses and co-infections are honestly miles above my ability to explain. But luckily, my good friend and collaborator Xie the Neutrophil has already made an incredible video that can take you through the topic. And here he is now with just a little teaser.
Whoa. Where the heck am I? Am I in the clockwork blender space? I'm not used to feeling so thick. Give me a second. Much better. That's right, folks. Hep B and Hep D, also known as the Delta virus, form what is possibly the weirdest dynamic duo in all of virology. Come on over to my channel to learn more, where we're going to talk about all the bizarre interactions that happen when you get infected with two viruses at once.
Okay, seriously, go watch that video now. I actually do make a cameo in there and explain hepatitis D in just a tiny bit more detail. But here, there's nothing more in this video. Quick, let's just abruptly cut to the outro music. And hey, thanks for watching all the way to the end of this video. Sorry for the abrupt finish here, but I really want to guarantee that you go check out my friend Xie the Neutrophil right now. Like literally right now. Why are you still here? If you like this video though, and you're still here for whatever reason, make sure you like and subscribe. Also, please comment on Phai's video and tell him that Clockwork sent you. It actually provides us a lot of invaluable data that we can use to plan future videos. Otherwise, I would also really appreciate it if you checked out patreon.com/clockworkshow to join our monthly sticker club or molecular papercraft club to support my channel specifically. But I'll catch y'all in the next video. For now, I like to leave you with peace, love, and nuclear localization signals. Everyone be well. Thank you so much.